Now, what happened to the central nervous system? It has to wait, this 1941, it has to wait for 30 years before we discover the long-term potentiation in the brain. High-frequency stimulation can actually increase the efficiency of transmission in the hippocampus, a very important for learning and memory. But in the hippocampus, the transmission is in 1 second high-frequency stimulation would result in the increased transmission, increased efficiency of transmission. And this discovery is also groundbreaking because this really tells you that in the central nervous system there's this activity-dependent changes that actually last more than 10 minutes, it lasts for hours. That's the striking thing. So it's called long-term potentiation. So if you don't remember anything about today's lecture, remember LTP. If you tell anybody you know LTP is, then you know neuroscience. This is most important keywords in neuroscience is long-term potentiation. A brief activity leaves a trace in your brain that lasts for a long time, called long-term potentiation. Now, if you have all the synapses gets activated, it can all be potentiated. Then in the end, you saturate all your synapses, right? So there got to be something opposite. And that opposite is found called long-term depression. Rather than have a high-frequency stimulation, if you have a low-frequency stimulation, in many parts of the brain, the activity, the efficiency drops. Now, this idea, you see this graph very often. If you just plot the magnitude of the transmission with before the experiment, in the control phase, you have a stable baseline. If you stimulate 100 hertz for 1 second, just 1 second, you get an highly increased response that lasts for a long time. In this case, 40, 60 minutes. If you instead of stimulating instead of 100 hertz, just 1 hertz, very low frequency, for 10 minutes, for a few minutes, the efficiency drops and then maintain at a low level. So this is called long-term depression. Now, according to the Hebb's learning rule, you can explain this. The Hebb's learning rule says if two cells are fired together, they wire together, they increase their synapse, right? When high frequency is firing, the postsynaptic activation accumulates with the synaptic potential accumulates, so the postsynaptic cell gets fired. So when you have the input of high frequency, presynaptic and postsynaptic cell fire together. So you get the first rule of learning rule, Hebb's learning rule. The second, low frequency, presynaptic cell is firing at low frequency, but it's too low, and each synaptic input are not capable of exciting the postsynaptic cell. So you have a presynaptic cell keeps active, postsynaptic are not active. So you don't have you have an uncorrelated activity. So the synapse gets depressed. Right? So that's a simple learning rule you can explain this. Right? So this is the LTP and LTD. Now what happened at the synapse? If you look at the synapse, each cell, each nerve cell receives thousands of input. Each of these tiny particles here is actually dendrite. The synaptic spines on the dendrites, they are the site for contact. The excitatory input to a nerve cell usually ends on these protrusions on the dendrites. Dendrites are the branches the cell send out to collect information. On these branches, there are thousands of these spines receiving individual input. Right? So it's the summation of all this input that the cell is integrating. Once they integrate and the summation is above their firing threshold, they may fire a signal. Right? So the cell is integrating information. Some of the input are not on the spine on the site, they are inhibitory synapse that gives instead of exciting the cell, they inhibit the cell, cause the potential to go down. So you have a positive and negative input, you sum everything together to decide whether you want to fire. Now why is there such a strange structure called spines? Why do you need the spines? Now here I want to bring again another very major important point made by a Chinese neuroscientist whose name is Zhang Xiangtong. right? John Shelton was a Yale trained. He went to Yale for PhD during the war. Was trained at Yale as a physiologist. He was the person who are the best in the early 50s in recording from single neurons. And he noted that on this neuroni, on this dendrite, there are a lot of this protrusion. So he proposed in a symposium in 1952 that this special structure protrusion are the site for controlling the input. But because you have a structure, then the current flows in will influence the cell differently if the structure is different. So you can use the structural change to control, as a gauge, to control how much current you want to flow into the dendrite. And that's weight control. The input, you can control its contribution to the cell in terms of determining its output by changing the size. Now this idea was buried there in 1952 until 30 years later, Francis Crick, you all know Watson and Crick, double helix. Francis Crick was the first one who dug up this idea. Francis Crick, who his solution of DNA structure and genetic code, after the genetic code was solved in the late 60s, Watson went to do cancer studies. Crick decided there's no more things about genetics that's important. The more important is the brain. So Crick became a neurobiologist. Now he's a theoretical biologist. He doesn't do experiments. He just thinks. He just sit there and propose hypotheses. Now one of the hypotheses he proposed is that he asked the question, does the dendritic spine twitch? So according to H.T. Chen, he said in the, let's see, well, this is what he said. The most interesting suggestion about the spines spring from a remark by H.T. Chen in 1952 that this structure is a controlling element for information flow. So suppose this spine can twitch, can change its shape. Then if activity can cause spine to twitch, then activity can change this information flow. That's his suggestion. It's a hypothesis made by Francis Crick. So people start to look for whether spine is dynamic, whether it actually can change. It turns out that they do change. So Crick prediction not always work out. This one is one that work out. Many of his predictions are actually hasn't been demonstrated to be true. The LTP, that's the potentiation produced by activity, right? You have a change in synaptic strength that goes up and persists for a long time. If you follow the spines on the dendrites, after 30 minutes after the LTP is induced, during this period, after 30 minutes, there's a new spine that forms. I don't know whether you can see well, but there are these arrows that point to new spines that appear after LTP inducing activity. Now this new spine means a new site for synapse. So there's new structure that can form. Now this spine can grow, and we are actually a few years later we show that spine can shrink. If you have a LTD inducing stimulation, some spine gets reduced in size, some will actually disappear. Now all these are in vitro experiments in brain slices. The brain slices are not a real natural condition, but you can really look at a high power. You can see detailed structure of the cells. You can do functional study. They are still alive in a brain slice in a good condition. You can study a lot of interesting phenomena about the brain, but it's not in vivo. So then we need to know what happened to the spines and synapse in vivo when you have a memory, right? So that's a question that now available last 5 to 10 years. The in vivo imaging of spine in high resolutions, you can stain the cell, introduce fluorescent protein into individual cells. You can actually see a structure. You can open a window on the head of the mice, with cells fluorescing underneath, you can actually see, see if you can see, different levels. You can go different levels into tracing the individual dendrites into the brain, a few hundred micron deep. Under dendrites, you can see individual protrusions, the spines. In fact, the spines you can see in vivo. You can see protrusion, you can see presynaptic axon comes in here and making contact with the spine, making synapses. So you can watch dynamics of synapse in a living mice and ask what happens to their changes. In fact, people look at adult mice. The first thing they show, we can compare 4 days and 2 weeks later, same region, same spine, compare the size and the density. You see whether there's change during this period of learning, right? Repeated observation on the same site. So this answers a long-standing question about what really happened in the connection. So when you have an LTP, you keep a memory of experience by changing the efficiency. But if the connection between two neurons are changed, you are talking about structural change in addition to the functional change. So how much is a structural change, how much is function change? And there's a long hypothesis over many years that when we are forming new memory, maybe we are changing our circuits, we're making new connections in our network. How much new connection are actually made in the brain, in an adult brain? So the answer to this, you can answer by looking at in vivo. It turns out that the spine turnover, what we call the increasing the spine and decreasing spine is a very small event, a very low frequency event. Most of the spine in the adult brain are stable. About 10% or less spine can form. If you keep every day observation, look at every other day, how many spines disappear, how many spines are forming, you find out it's always below 10%. The majority of the spine and the presynaptic structure remain unchanged. And even during development, there's some change. In early development, 1 month old, you have a higher level of illumination. Lower level, the illumination is high, but 2 months later, the percentage of illumination is lower. But there's a constant level of formation and a constant level of illumination. So there's a basic change in the connectivity. So the question is, what happens when you have memory? You really form a specific memory. What happened to the, does it change the percentage, the rate of formation of new synapse, or the memory is actually wiping out old connection, formation of new connection?